brief introduction of EMTech will be delivered by Head of EMTech, Universiti Putra Malaysia, to Professor Syukuri. The time is yours.
Well, thank you, Mr. Chaps, MC. So again, thank you again to Pak Fatani and team from Applied Research Center of Applied Microbiology for again having us, inviting us to come here, and of course, wonderful to be here again for me, and brought along my team. Hopefully, in this few hours of session, we should be able to get some ideas and share knowledge and research findings to all of you who are, of course, must have some at this moment interest into some structural biology. So before our colleague Dr. Fiza and Dr. Dina, and of course the other speakers, Dr. Dede and Dr. Joko presenting their work. So let me just allow me to introduce our research center. So we are coming from Enzyme, from UPM, of course, in Universiti Putra Malaysia. To those of you who've been to Universiti Putra Malaysia, about 30 minutes south of Kuala Lumpur. Okay, so easily accessible through railway networks and of course any other modes of transportation. So we are working in the Enzyme and Microbial Technology Research Center. In short, we call it EMTech. It is very much one of the key important research group in the country, where we started off back in 1986. Okay, back then started off as a research group, and in 2012, the research center research group was promoted to the research center of excellence by the university. So our research very much focused into two keywords over here: enzyme. Working with enzyme, and of course with microbes. So most of the enzymes come from microbes isolated from local isolates and some other parts of the world. Okay, correct. It's not changing. Okay, right. Sorry. Okay. Yeah. Sorry, I think. Okay, all right. Okay, so that's me, basically currently heading the center. And of course, together with me at this point, we have our EMTech research advisors, Professor Emeritus Dr. Waqar Saleh, very much a prominent enzymologist in Malaysia. Also, we have, of course, Prof. Raja Nur Zaliha Raju Abdul Rahman, where her expertise is more to molecular biology and structural biology, where she started off the structural biology crystallography project in the country back then when we started off in the project back then in 2004. Okay, so and together with us, a number of molecular biologists and structural biologists by training. Dr. Adam, he couldn't make it, of course, with Dr. Nomi as well. So basically all of us, all of them are coming in the background of structural biology. And you also see over there is Dr. Surana Sabri. So more to expertise on to material engineering. Okay, utilizing E. coli, for example, to modify their pathways in order to absorb for the E. coli to consume sugar. Okay, so next we have also with in the team a bioinformatician as well. So Dr. Siti Nobaya Oslan. So she's currently doing sabbatical leave, but she unable to come with us. And also Dr. Fairuliza, another structural biologist. So more to looking into enzymes in case of lactase, okay, and some other inhibitors for alpha amylase inhibitors. She's graduated from Universiti Putra Malaysia. And you have also here Dr. Nafiza and Dr. Nodina Mamano. So our two speakers for today, they have, of course, some vast background in terms of structural biology as well, and very unique if you look into Dr. Dina's background, the field of picobiology, okay, from Japan. So just structural biology by looking into the atomic scale at picometer level. Okay, so we have also in EMTech, we have a postdoctoral research fellow and a research officer, and also brought along Ms. Nik Fauzan, Nik Muhammad Aman as our Assistant Science Officer, okay, in working in our EMTech. Okay, and generally, we EMTech research more emphasize on the microbial technology, microbial and enzyme technology. So more or less, we want to explore and see and what sort of capacity of this organism to produce such enzyme. We have worked with some extreme enzymes, for example, those coming from the extreme cold, okay, from enzyme from the South Pole, Antarctica, from the hot lakes, okay, and of course from our own backyard, palm oil mill affluents. So this enzyme, these microbes are able to grow at high temperatures, okay, from thermophilic in terms of thermophilic microorganisms, and we want to see what their enzymes that they are produced. These are, of course, enzymes that are industrially relevant, and of course beyond that, we want to see into the fundamental aspect of this enzyme. Okay, how does this enzyme actually works? This the mechanism, the mechanistic activity of this enzyme. So in terms of utilizing bioinformatics and plus with the now current ultimate structural biology, we call it, okay, the crystallography. And very fortunate that BRIN has acquired very powerful structural biology tools, okay, called the cryo-EM microscopy. So this, I believe, shall be a process. I mean, shall one way to accelerate further the development of structural biology in Indonesia. So crystallography has been one of our core research. Okay, so we started off back in 2004, where we produced our first crystal in the country, protein crystal. So most of which the protein that produced as a recombinant, recombinantly. Okay, so we use yeast and E. coli as the workhorse for the production of this recombinant protein. Okay, so generally, what we do in EMTech is, of course, start off with enzyme isolation. So students in the lab typically will have the opportunity to start work from enzyme isolation of microbes. We'll do some molecular biology, perform some in silico and computational biology, and towards the end, in the end of their work as well, looking into these crystallographic structures of the enzymes and protein. And to some extent, some of the students do also perform, some scale-up studies utilizing bioreactors. Okay, we have small-scale bioreactors, 7 and 30 liters in scale. Yeah, but so it is the merit of information, new knowledge to be taught to the students of interest working in EMTech. So you may see over here that the protein that we work, typically most of the protein we going through some structural biology studies, but again in order for us to run some crystallization experiment, protein purifications and expression is very important aspect to be considered, okay, because production of enzyme as recombinantly sometimes may adhere some additional issues, okay, it comes to the case of misfolded protein aggregations and many more. So, and of course, we also utilizes computational biology. So this basically helps us to to complement the data that it is obtained from the experimental processes, crystallography, NMR, and to couple with this computational and in silico approaches. So we should be able to get some additional insights into this protein structure folding and mechanism. And so this is looking to aspects onto the fundamental. We have also studied on some antimicrobial peptides and proteins, okay, isolated from our own local isolates. So these peptides and proteins are very much very important in inhibiting microorganisms related to MRSAs, MRT-resistant Staph aureus, and some of which is trying to work with some managing our diets, controlling our sugar intake by inhibiting alpha amylases, okay, utilizing palm oil, I call it proteins and peptides coming from palm oil sources, okay. So very quick of enzymes and proteins that we have studied and still ongoing. lipases and esterases at the top left of the slides are one of the earliest enzymes to be studied. Still in 1986, we started off working with lipase and esterases because the project back then started off with looking into this, the potential of palm oil mill effluents. So of course there are a lot of bacillus growing there, thermophilic bacillus. So we found also a number of lipolytic enzymes that now still should very much industrially important, considered to be very important industrial enzymes. So of course the list goes on, and we have also Dr. Hafizah and Dr. Dina working into plastic degrading enzymes, PTAs, PLAs, and also enzymes and that are involved in productions of fine chemicals, biodiesel and biofuel, in the case of alcohol dehydrogenase and the carboxylate reductase, right? So, and we have also studied some, of course, some new proteins, starts to emerge into our catalog in terms of the study of aquaporin and the aquaglyceroporin. So these are proteins that are very much prevalent in all living organisms, where it's a protein that's responsible to transport water molecule, in and out. So we have them a lot in our kidneys. So this has a lot of potential impact in terms of, let's say, for example, biomimetic water filtration membrane and the likes. Okay, so all of this, some of the enzyme has gone through a lot of structural biology aspect, looking to their structure and functions, but majorly utilizing using the modes of protein X-ray crystallography. Okay, next. Okay, so very quick info about our linkages. Very important that I think what we are doing now with BRIN and with this research center of Applied Microbiology. So we hope these collaborations tends to be more will be more productive, because current research trend is looking to more not as a single disciplinary research, but we are looking into multidisciplinary and transdisciplinary research. Okay, in the end of the day, the research has to be conveyed and to be translated for the sake of the community out there. Okay, so we hope to benefit them the most in the end. So a lot of which universities, of course, some of you may have been to some of these major universities, Universiti Malaya, UKM, USM, so some of which even more are considered alumni, maybe some of you. So these are some of the top national universities that we collaborated with, and also with some Petronas, just like here we have Pertamina, for example, your petroleum company. We also work with them and some of the major plantation companies. Okay, palm oil as one of the major cash crops in the country. Okay, next. And yes, not just being have network internally. We also have linked to some of the major what you call this research institutions and facilities to have access to world-class facilities. So and probably you have heard about Spring 8 super photon ring 8 gigahertz electron volts. So these are electron synchrotron facility that we actually use to help us to get information, more detailed and accurate information about protein structures. Okay, so this has been made success with the collaboration with Osaka University, specifically those from Institute for Protein Research. Okay, very well known, well reputable research institute in the world. And we have also worked with Spanish Science Research Council, looking into enzyme immobilizations, enzyme reactions and catalysis. And of course, some of the highlights again on the crystallography is JAXA, Japanese. We have also experience working with protein crystallizations and microgravity in space. Okay, where we send protein to international space stations to see how is the behavior of protein in space, and for our case, we want to see the crystallizability of that protein in the at zero gravity environment. So we wish to compare how is it in the terrestrial environment and to the zero gravity, and yeah, of course, we find something very interesting out there. Okay, the protein behaves differently. So one aspect that we feel very fundamental research in nature, But that small findings actually has a lot has a major impact when it is implemented on Earth, for example, in our situation. Okay, hopefully, inshallah, BRIN shall be will and continue. Hopefully, our partner. So a lot of similar terms in terms that we similar interests. Okay, that we see. I think ever since the visit from Pak Watani and delegates in 2003. So we have trying to forge stronger collaborations, and in terms of many aspects, one of which is the research by degree, for example, at UPM. Okay, that's one aspect that we intend to work with in the long run. Okay. Probably not talk much about this one. Maybe I have additional slides actually, not presented this morning. Okay, next. Sorry. Okay, this one probably I worth highlighting as well. So in doing a research, of course, being an academics in universities, so publication is very important. So our students has been very, alhamdulillah, very productive. We have able to get our work published in quarter 1 and quarter 2 publication, and this is the rank is based on Clarivate Analytics. Okay, what Web of Science, probably you have heard it before. I think you are using the same database, I presume. All right. So this database has been used by universities all over the world on top of Scopus. Scopus will have their own ranking. So we have additional rank used by our university, particularly those that is categorized under research university. So UPM is one of the university that is categorized as called as research university in the country. So we have 5 of them in it. Okay. So of course, students graduated on time and are able to produce such papers has been tremendous to us, right? So, yep. I think next. And filing intellectual properties, protections of ideas and innovations is also very a practice that we applying. Okay, protect the ideas even before the work is being made for publication. So we have number of files, patents, and granted at the same time where it is submitted. Okay, and granted in various countries. Okay, United States, Europe, Japan, and the likes. And some of the additional copyrights, I think probably you know, and in terms of intellectual properties, they are copyright, trademarks, patents. These are types of intellectual properties that we are of course interested with. Okay, so hopefully in the end, talking about commercializations later on. So this shall be one aspect if you want to go to licensing and many more. So our laboratory is very small, unlike BRIN. So BRIN is we are very impressed with all your facilities. So, but of course, the lab is very much scattered throughout the campus. We, of course, some are located in Institute of Bioscience. We call it in Biotech 2, Biotech 3. These are located in our Faculty of Biotechnology and Biomolecular Sciences, right? And in the lab, we have facilities that are devoted into looking into these, our activities running as smooth as possible. Okay, next. So we have these facilities. I think some of which some of you are very much in common with, but probably the bottom 2 is quite unique for crystallography. We instead of using a human power, of course, learning from basics manual is very important before we let the robot to do the job. Okay, Oryx 8 crystallization robot to help us do all the dispensing of solution and making up, prepare the crystallization droplets. It is not just 1 or 2 droplets, can be hundreds and thousands of droplets. So to minimize the human error, we employ this robot to help us, make the work even faster. Okay, while single plates may require about 2 hours, the robot can do it in 50 minutes, for example. Okay, so this crystallization process, for example, by Proton, our speakers may share to you later on, we will involve in the process of crystallization screening. Okay, crystallization screening is to have us to improve our chances in getting crystal hits. Okay, so we do 1 or 2 types of crystallization. We do thousands of screening. Okay, because not all protein, bear in mind, not all protein can be crystallized. All right, so as of today, maybe 30%, 40% proteins are crystallizable. All right, so that is considered to be one good success rate, I think, globally So, but now with the techniques has been more advanced and developing, more and more new screening solution has been developed to allow other proteins that is difficult to be crystallized. Okay, right? So next, right? Yep. Some of the facilities, dichroism spectropolarimeter is one of the simple tools to measure, to measure the protein sequence structures, and using polarized light. Okay, and of course, some of which Acta purifiers. You have more wonderful units like this. This one has been more than 20 years, I think. Still running, still running, and most of the work has been done do it yourself, DIY. So we do the maintenance ourselves. Okay, right. That's in general. So okay, next. So in terms of modern structural biology nowadays, we are talking about utilizing all these tools, not just cryo-EM, X-rays, all these additional techniques is actually is made available. Some of which are even in BRIN, so we can have this data be complemented, okay, to add value to the research finding that we have. Okay, so but it is not a must that we must all go through all this. Okay, just to have cryo-EM and crystallography. These are what we call it the two ultimate structural biology tool that will give us the sub-angstrom resolution, okay, atomic scale information of your protein structures. Okay, so basically, of course, some of the pipeline looking to the diagram over there. So you can see from isolation, expression, purification, crystallizations, antiloop structure. So that is considered to be one cycle in order for, let's say, a PhD degree. Okay, you can graduate as a PhD degree having that downside, having to be able to complete those tasks. Okay, yeah. Okay, so just again, I mentioned about our collaboration with our international partners, one of which is the Japanese Space Exploration Agency and NASA. So of course, we have the opportunity. We were given the opportunity to send protein to international space stations. So we started off back in 2007, where we launched our first astronaut to space. So he had some 8 days there of spend his time at the International Space Station. So he brought along many other missions: seed to space, worm to space, food to space, including a protein to space. So that was one of the key highlights in terms of structural biology project, not just for EMTech but also for the country in general. So then subsequent years, Japanese team approaches, and then more and more missions. 6 additional space mission was sent through the we call the Russian space agencies, and we have about 20 plus protein was sent to space, and about 18 of the structures were successfully solved. Alhamdulillah. So we don't get this opportunity very frequent. So it is not a must for you to send crystal to space, but it will be a good idea if you have the capacity to send, because it is everything can happen up there. So which is limited information can be gained from that such experiment. Okay, next. Just a picture again, our first protein crystal in the country. This was produced by Roger and his student, Dr. Adam, actually. Adam Rutianchu is her student, including me. I am her student as well. So, so this was back in 2004, I think 2004, the first crystal. Lipase, this is a lipase, by the way. Okay, from thermophilic bacillus, Geobacillus T1. Okay, so it is small. It is about 0.2 millimeter in size, but this one, the size of the protein is about 42 kilodalton. Okay, so crystallography has limitless in size, so you can from this big size of megadalton to the small size of kilodalton. Okay, so but only that the bottleneck, the challenge will be on the crystallization aspect. For the cryo-EM, there will be some added advantage is that you can work with protein in solutions. So that will have you will be able to get some information about how the dynamics of protein in solution. Okay, that is very strong information that we can actually obtain. Okay, next. All right. Beyond that, more and more crystals were produced in our lab. Okay, some crystals for me is very nice, wonderful, nice looking crystals. I hope you can appreciate the photo very well. So we took them under polarized light. So a certain angle, the protein will exhibit colorful images. So what happened in our lab, we take the photo and have them printed, and posted in the frame in our lab. So yeah, so before this is before the crystal is being after X-ray damaging, being damaged by X-ray. So it has to be taken before, and this is how it looks. Very small again. 0.1 to 0.2 millimeter is one of the ideal size of crystal that we actually obtain in our lab. Okay, I think that's all as far as the structural biology and EMTech is concerned. So I hope today you're going to have some wonderful seminars, and yeah, if you have information, person to contact, yep, you can just reach us at any time soon. Thank you. Yeah.
Yeah. Okay. Thank you so much, Prof. Shukuri, for the brief introduction of MTech. Okay, I think everyone cannot wait longer for the seminar because everyone is ready. So without further ado, let's start our seminar with the resource on protein structure and engineering, and will be guided by Dr. Linda Sukmarini. Please give a warm round of applause. Okay, Dr. Linda Sukmarini, the time is yours.
Okay, thank you, Ms. Dea, for bringing us to a very productive joint seminar today between EMTech and BRIN. Good afternoon, all. Assalamualaikum warahmatullahi wabarakatuh. Honorable Head of Research Center for Applied Microbiology, Dr. Ahmad Fatoni, and Honorable Head of EMTech UPM, Prof. Dr. Muhammad Sukuri, our distinguished speakers from UPM and also from BRIN, and all delegates from EMTech UPM. Welcome, and of course all audience online and offline from BRIN or university. Bapak and Ibu, I'm Linda Sukmarini. It gives me a pleasure today to be a moderator for this joint seminar. As we know that for decades, protein engineering has been utilized to modify the natural protein and enzymes to meet the needs of different industrial applications. And by elucidating the protein structure, it can be modified via protein engineering. Therefore, the structural biology take part in this role. And in this today's seminar, we will get a closer look into structure-driven protein engineering for production of valuable product for industry. And first of all, I would like to welcome you for this presentation. First, we have the two sessions. First session, we have two speakers from EMTech UPM. EMTech delegates will present about, I think, protein engineering and structure biology elucidation. So first, I will welcome for the first speaker, Dr. Nur Ahmad Hafizah Ahmad Komarudin. Please welcome. So please show the CV. Okay. I will read. Wait. Wait. Okay. Let me first read the CV of Dr. Nur Hafizah Ahmad Komarudin. He's a senior lecturer at EMTech UPM. She graduated from Microbial Biotechnology from UPM and is now active researcher, a senior researcher also at Enzyme and EMTech. And she committed member of Protein Science Society from Malaysia and many others science community in Malaysia and international community. And her interest particularly in structure and function study of industrially relevant enzyme, especially hydrolysis, and then many others protein engineering actually on enzymes, yeah. Okay. And without further ado, please welcome to your presentation. Yeah.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon. Thank you for the kind introduction, Dr. Linda, and thank you, BRIN, for organizing this session. I am Nur Hafizah Ahmad Komarudin, one of the EMTech members, and I'm delighted to share with you some of our research findings on protein engineering and structural modification. So the title of my presentation today is Engineering Strategies to Enhance Stability of one of the industrially important enzyme, which is lipase, and I'll be focusing more on staphylococcal lipase. Before I start the presentation, this is just a quick view of my research areas and my current research focus. So I would like to welcome an open collaboration with BRIN researchers. I'm doing on microbial biotechnology and molecular and structural biology. So my current research focus is on structure and function studies of industrially important enzyme, lipase and alcohol dehydrogenase. And I do X-ray crystallography, and we use also computational approach to study the enzyme. And I also do some engineering studies for enzyme to improve the properties and also for structural analysis and understanding. And for the microbiology work, currently I'm doing some identification of bioplastic degrading bacteria and also some plant growth promoting bacteria. Okay, now back to the presentation. Lipase is one of the most industrially important enzyme. It is very popular. It has been studied for many years, for many decades. But the real challenge is to find the enzyme that can be used or applied in industries, because for industrial enzyme, there are certain criteria required. For example, the enzyme needs to have high activity, high stability, is in large scale production, and the enzymes have to be resistant to denaturants. So given the vast reaction conditions and diverse industrial requirement, so it is not always easy to find the correct match of the lipase or any enzymes, yeah, to the industrial requirement. So and furthermore, because there is a poor understanding of the enzyme structure and function, so it limits the applicability in real industrial settings. Okay, so to overcome the limitation of natural enzymes, protein engineering, to overcome the limitation of natural enzyme, protein engineering was developed many years ago. So in general, So in general, protein engineering is divided into three, can be divided into three: directed evolution, semi-rational design, and rational design. And these protein engineering studies has seemed to evolve from time to time. I recently came across one paper about machine learning, because we have now AI, so machine learning in protein engineering. So during the years where there is very limited number of protein structure, directed evolution was mainly used to design or to engineer protein. But with the rising numbers of protein structures in protein data bank, we have more knowledge now on protein structures. So we can use the protein structures to do semi-rational design and also rational design to find or to mutate a specific mutation site. For the case of lipase, where is my pointer? Okay, wait. Sorry. For the case of lipase, for the engineering study, the work has been, many works have been focusing more on improving the enzyme properties, for example, thermostability, organic solvent stability, and also substrate specificity, but not only limited to the improved properties. Engineering of lipase is also used to study or to identify certain residues, important residues that is responsible for the function of the lipase. Next. Okay, so now I'll be talking about staphylococcal lipases. So staphylococcal lipases is one among the lipases that have potential application in various industrial applications. So staphylococcal lipase is a true lipase, and it is classified in a different subfamily of lipase because of its unique characteristics in terms of, one, the molecular organization, where it has the signal peptide, propeptide, and also the mature sequence. And staphylococcal lipases are also phylogenetically distant from other bacterial lipases, where they share only 30 to 40% identity with other bacterial lipases, for example, Pseudomonas lipases and also Bacillus lipases. And they are equally important as other bacterial lipases because there are many studies conducted and have shown that these staphylococcal lipase also have interesting biochemical properties. And in terms of structure, there is a very limited knowledge on the 3D structure available in the protein data bank. But interestingly, the structures that have been solved are in open form. So lipase is known to have this lid, because this is a true lipase. It has the lid structure, and in the case of staphylococcal lipase, the lipases have been solved in the open form, means the lid is open for substrates entrance. Okay, next. Okay, so what have been reported so far for staphylococcal lipases? So staphylococcal lipases have been studied since 1985. The first one was from Staphylococcus hycus lipase, and there are many other lipases reported from different Staphylococcus species. They have been characterized, biochemically characterized, purified and biochemically characterized. But in terms of structure, we can see there is still a gap in understanding the structure of staphylococcal lipase. So the first structure deposited in protein data bank was Staphylococcus hycus lipase in 2007, and we can see a huge gap between 2007 until the second structure was reported in 2020. And the second structure deposited is from Staphylococcus aureus lipase. So in 2020, there was another lipase reported, but it is not really stated whether this is a lipase. It is generally stated that it is a serine hydrolase. And in 2024, there's another one, but it is coming from the same lipase, which is the Staphylococcus aureus lipase reported in 2020. Okay, so this is the enzyme that we have been studying. The lipase from Staphylococcus epidermidis 82. So this enzyme is produced by an organic solvent tolerant Staphylococcus epidermidis 82 that was locally isolated from a car service area. So the motivation during this time was to search or to isolate organic solvent tolerant bacteria that produce organic solvent tolerant lipase. And we express it, we purify it, and then we characterize the enzyme, and we found out that this enzyme has two unique features. One is it is a co-adaptive enzyme, which is active at 25 degree and stable at temperature below 30 degree. And it is also stable in polar and non-polar organic solvent. So these two are interesting features for application in low temperature processes. So it can reduce energy consumption. Because there is not much information on the structure, so we were keen to study the enzyme structure. So we attempted to crystallize the protein. We managed to get a few hits, but unfortunately the crystal hits were not reproducible. So we did many strategies to improve the crystallizability of this enzyme. We did large-scale expression. We modified the method of purification. We even construct new clones to get a better or soluble expression of the enzyme. And we did massive crystal screening. But unfortunately, this enzyme was found to be recalcitrant or to crystallization. So for stubborn, we can call it as stubborn crystallizers. Many studies reported that probably we should look into the structure of the enzyme. So what we did next is we predicted the structure of 82 lipase with the only one available structure. And then we managed to get the structure, and then we superimpose the structure with the available structure, and we try to find any dissimilarity between the structure. Okay, and then we found that this region, the one in circle, probably affect or provide the flexibility to the enzyme structure, and probably contribute to the difficulty in getting the protein crystals. And we did sequence alignment, and then we found out that the last four residues intrinsically disordered region based on X-style PRED2. And when we observe further in the structure of Staphylococcus lipase, we found out that the last two residues are not even in the structure. So what we did next is we add a stop codon, just a simple structural modification. We add a stop codon at the C-terminal end of the enzyme. So we remove 4 residues, and then we run simulation to check what happened if we remove the C-terminal end. And this is one of the analysis from the molecular dynamics simulation on the RMSF values. So the one in the circle is showing you that the C-terminal end has a very flexible, is very flexible region. But when we removed it, we can't see any any increase in the RMSF value. Next. Okay, so that is on the molecular part, and then we purified the enzyme, we characterized the enzyme, and we checked on the effect on crystallization and other biochemical properties. And interestingly, alhamdulillah, we managed to get the crystals, but there was another challenge because X-ray crystallography is not a straightforward process. So we managed to get the crystals, but we couldn't solve the structure because of the poor diffraction spots. And then for thermostability, because we removed the flexible terminal end, so we wanted to know what happened to the biochemical properties. So interestingly, we found out that the mutant with the truncated C-terminal end exhibited higher stability compared to the wild type. The mutant is shown in gray color. And we also studied on, or we also examined the homogeneity property of the enzyme, because this enzyme was not easy to handle. It had many problems in terms of it is very easy to degrade over time. So when we removed the C-terminal end, we found out that it has improved the homogeneity of the enzyme by looking at the native page. So in conclusion, for the first study, deletion of C-terminal end yielded a mutant with improved crystallization, but of course further optimization is required for structure determination. And we managed to improve the thermostability profile, and also the mutant has improved homogeneity. Okay, and then the second study on protein engineering to improve the property of this enzyme, we did directed evolution of the previously truncated Staphylococcus epidermidis 82 lipase. We did error-prone PCR. The aim was to improve the thermostability of this enzyme, hoping that the enzyme has broader temperature profile. So we did error-prone PCR, we did screening, we selected from the mutant library, and we did the expression at 50 degree. And we managed to get one mutant with 4.84 increase in the activity at 50 degree. So we selected this enzyme, and then we further analyzed based on qualitative analysis. If you look at here, there are three plates showing you that the wild type has no activity at 50 degree, but for mutant, there is this activity. And then we did sequence alignment, and then we found out that there is a mutation, but there's only the mutation happened at position 210, where glycine was substituted to cysteine. And then interestingly, because this enzyme has no cysteine in the sequence, so random mutagenesis has introduced single cysteine in the sequence of this 82 lipase. And then we further investigated what happened to the structure to the enzyme, and we further confirmed the thermostability profile. And if you can see the graph here, so the wild type is active at 25 degree, but we managed to improve the optimal temperature of this new mutant G210C by 20 degree Celsius, in optimal temperature. And we did some thermostability study. We did some thermostability study, and we proved that the new mutant has is able to retain its activity at longer time at high temperature. And we check also the midpoint of thermal unfolding using CD, and we could see that the enzyme, the mutant, has a 10-degree higher TM than the wild type. Other parameters are comparable to wild type. And then, yes, next please. And then we further study the enzyme structure because we wanted to know what make the enzyme thermostable. So we look closely at the site of mutation where it is located at the loop of the two lids, and then we did the mutation MD simulation, and you can see the blue color spectra where the mutant has improved stability and also improved rigidity in terms of the compactness of the protein. The more stable the protein is, usually the structure is more compact compared to the cold enzymes that is more flexible in structure and less rigid or compact. Okay, next. And then we tried to identify or investigated on the, examine on the intramolecular interactions. So we compared between the wild type and also the mutant. And interestingly, in the mutant, there are additional salt bridge network, increase in hydrogen bond number, and also there are more cation pi interactions in the structure. I didn't show here. So that all the intramolecular interactions may contribute to the improved thermostability and also improved rigidity of this enzyme. And for the local structural change, we can see there is a shift in the orientation of this glutamine, and the glutamine has formed two intra-residue bond and compared to the wild type. So from this study, we conclude that random introduction of a cysteine residue at the loop bridging the two loops likely stabilized the lead and provided overall rigidity to the structure of the mutant lipase. And then for the last engineering study on this enzyme, we did site-directed mutagenesis of surface exposed lysine because we were trying to find out what causes this enzyme to be difficult. Why is it difficult to be crystallized? And then we look into the structure, and then we found out that there are many lysine on the surface of the enzymes. So there are studies reported that when there are too many hydrophilic or large residues on the protein surface, it may interrupt the crystal packing and also the crystal growth. So we showed the structure, we predicted the structure again, and then we can see that the surface is full with lysine. And interestingly, when we compare the proportion of lysine between staphylococcal lipase and other bacterial lipases, staphylococcal lipases have many lysine in the sequence, and most of the lysines are conserved, but not for bacillus lipase and pseudomonas lipase. So we listed all the lysine in the sequence, and we analyze in terms of the SASA value, the solvent accessible surface area, secondary structure, and whether the residue is buried or exposed. And we further use a prediction tool to rank the residue because we want to identify which residue of lysine to mutate So we don't mutate all because it will take time to mutate all. Okay, so we based on the prediction tool, so there are three lysine that was targeted. So 91, 177, and also 325G. And from the single mutant, we also randomly combine the single mutant to form another three double mutants, 91, 177A, 91A, 325G, and also 177 and 325G. Okay, and then before we constructed the mutant, we did in silico analysis of all the surface lysine mutants, the 6 surface lysine mutants, and based on this in silico analysis, we managed to identify stabilizing mutant and also destabilizing mutant. So when we look at the simulation results, we found out that mutation at 177 is a destabilizing mutant where if we mutate to alanine, it is the structure become disrupted or become less stable, and we further analyze what is between this residue, and we found out that this residue is conserved in the sequence, and also it actually forms bonds with other neighboring residues. So, but not for the case of 91 and 325. For lysine 91 and lysine 325G, when we run simulation, we found that these two mutants have improved stability and also rigidity. So we proceeded with the experimental work. So we constructed the mutants, and then we expressed it, we purified, and we did the biochemical characterization. So based on the biochemical characteristics here, we actually tested all routine parameters, for example, temperature, pH, substrate, but the one listed here, just the summary. So for temperature, we could see that if we mutate the 325, we mutate lysine at position 325 to glycine, so it increased the optimum temperature of the wild type by 5 degrees. But for the double mutant, it decreased the activity by 5 degrees. So there is a variation in the biochemical properties of the mutant and the single mutant and also the double mutant. And you can also see there are other variations in the biochemical properties between the three. And we concluded that the replacement of lysine with a smaller amino acid may result in both stabilizing and destabilizing effects. And even if we combine the two stable mutant based on molecular dynamic simulation analysis, it could also accompany with this stability and activity trade-off. So we can see a mutant with destabilizing effect and also with stabilizing effect. Okay, so the conclusion for the three engineering studies. So the first one on the deletion of the terminal ends or the flexible region. So it contributes to stabilization of the enzyme structure as well as improved properties. And for the site-directed mutagenesis, where we mutate the surface exposed residues, so we prove that even if the residue is on the surface, it is also important for the enzyme stability. And lastly, for random mutagenesis, we concluded that, yeah, it is actually a very useful approach to enhance enzyme properties without knowledge for the enzyme structure. Okay, so I would like to acknowledge my cikgu, all my teachers in front of me. So Prof. Raja, Prof. Bakar, Prof. Mahiran, Prof. Shukri, Dr. Adam, and these are all the project members. Yeah, Dr. Malihe, other EMTech members here, Dr. Dina, Dr. Farul, Prof. Dr. Su, Dr. Nurbaya, and my students who are involved in this study, Jivita, Nurul Nadira, and Ong Shani also she did some work on this 82 lipase, and of course to all EMTech postgraduate students. And I would like to also thank UPM for providing the support to do this project. So these are some of the references. If you are interested in our work, you can have a look at our papers. And I also included some of our research recent publication on protein engineering by other EMTech members. So yeah, that's all. Thank you very much.
Okay, thank you, Ms. Nur Hafiza, for your nice presentation. Okay, they're all. Before we move on to the discussion panel session in this first session, first we will go to the second presenter, Dr. Nur Dina Nur, PhD. Before I will read her CV briefly. So she's also the senior lecturer at Department of Biochemistry in EMTech, also at UPM. She did PhD in Microbiology, University of Hyogo, Japan. And I think she already published many journal for structure analysis, protein engineering also. And first, I will welcome to Ms. Nurdina Nur for come to the stage. Okay. So I will let you to present this year.
All right. Okay. Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Okay. Thank you very much, Madam Speaker, for your kind introduction. Okay. All honorable guests today. Thank you for spending your time. Okay, to be with us today. All right. So I'll be sharing my project, okay, my project presentation, some of the project that related to the mostly on the structure function studies. All right. So I'll be sharing some projects that related to the structure biology. So my title today for the presentation is the integration of X-ray diffraction analysis and computational approach to solve and understand the 3D protein structure. So in my project that I will be presenting today, so mostly not really, I'm going to present like 2 to 3 projects that combined. One of it is the by using X-ray diffractions and with the combination of computational, and the other 2 projects is mostly related with the only computational approach. Right. So next. Okay, next one. All right. So this is the overview of my presentations. So the overview, these slides, I try to introduce actually my expertise and the scope of my project that I'm involved in. So basically, my expertise in protein purifications, okay, and protein crystallization, protein X-ray diffraction and data analysis, and computational structure determinations. And I've been working with the enzymes like hydrogenase. I'm not sure how many of you may perhaps you're not really familiar with hydrogenase, but yeah, this is one of the enzymes that I'm working on. I've started the hydrogenase studies during my PhD project, and now I continue again for my students for the hydrogenase. And I've been also working on the serine and subsilicin and creatinase and metalloprotease, and explore its characteristics and enzyme potentials. All right, so next. So the first project that I'm going to share is about the structural chemistry of oxygen stable nickel ion hydrogenase from the Citrobacter species S77. So this is the project that actually the project that I performed during my PhD studies. So I'm going to share with you today about the finding of my studies. Right, so next. So when we talk about the structure determination by X-ray crystallography, or if we add on the another approach, which is a computational approach. So this is basically the method that involved. Okay, so it's not like straightforward. You get the crystal and then shoot the crystal and get the 3D analysis. Okay, so you have first, the first thing first is you have to get the purified protein. Okay, so highly purified proteins, so that you won't have big problem. Okay, in the other process later. Okay, and then in case of my studies, I'm also involved these composition methods, which is ab initio calculations and molecular dynamic simulations to confirm a few molecular species in the structure. Okay, so next. All right, so what is nickel ion hydrogenases? So I think just short introduction, because I think maybe some of you not really familiar with what is hydrogenase. So the hydrogenase is the hydrolase, okay, not oxidoreductase, okay, oxidoreductase that catalyze the simplest reactions, which is it catalyze the hydrogen, okay, the molecule hydrogens to very simple atoms, which is two electron and hydrogen. And then usually the standard nickel ion hydrogenase, they have a very high hydrogen oxidation catalytic activity, and it is mostly oxygen sensitive. Okay, so this is the standard nickel ion hydrogenase. And when it is really easily oxidized, okay, and becomes inactivated in the presence of oxygen, but if they are exposed to the hydrogen, okay, so they will be activated. Okay, okay, so the next one. So this is the overall structure of nickel ion hydrogenase that has been solved by previous researchers. Okay, so this is the standard nickel ion hydrogenase. So the active site of this hydrogenase, they have this X bridging ligands, which is connect between nickel and ion. So the state of this hydrogenase is determined by the presence of what types of X. Okay, so if X is there is a presence of hydroxide or OH minus, so they will become in the inactivated form. And if it's hydride or hydrogen in these positions, they are in activated form. And the other distinct how to differentiate, okay, the hydrogenase is between if they are actually the oxygen sensitive or oxygen tolerant, is by looking at the proximal cluster. Okay, the proximal cluster of this hydrogen. So basically, for the standard or sensitive nickel ion hydrogenase, they have a cubine type of the proximal nickel ion, and then for the oxygen tolerance, they differentiate with the formation of deformed iron sulfur cluster of the proximal part of the structure. Okay, so that is the difference. All right, so now we are talking about the subject of my studies, which is the nickel ion hydrogenase, which is isolated from Citrobacter species S77. So this was isolated from the national park in Kumamoto. It grows well in aerobic and anaerobic condition, and these are the characteristics of the hydrogenases, where it shows higher hydrogen oxidation activity compared to hydrogen production activity, and it has the optimum temperature for hydrogen oxidation at 65 degrees Celsius. So it's quite high. And all right, so the next one. All right, and one thing about the oxygen from CS77 is that it has high similarity with oxygen sensitive hydrogenase, which is E. coli HYDE2, which is about 93 and 95% for its small and large subunit. But by looking at the characteristic of CS77, it shows oxygen stability, okay, for like 100 hours. So the residual activity left is just 87%. There must be some questions. What makes this nickel ion hydrogenase from CS77 is oxygen stable compared to its other family members? Okay, so E. coli HYDE2. So we want to find out what makes this SM7 become oxygen stable compared to its homologous members. Okay, so the next one. Okay, so far for HYDE2 hydrogenase, there's no structure has been elucidated for HYDE2 type hydrogenase. So this becomes a motivation for us to find out the candidates for hydrogenase that oxygen stable, because we know that hydrogenase has received so much attention because it can be really high potential, okay, as the enzyme-based hydrogen fuel cells, okay, to and it's also a strong candidate for the next generation clean energy sources. Okay, the next one. Next, please. Okay, so this is the process or the method that I've done. Okay, so throughout my studies. So the first thing first, we have to get the as much the prefect protein in this case, and then get the protein crystals, and then diffract the protein, solve the 3D structure, and then in the meantime, during the getting a good structure analysis, so we have to adjust or analyze the electron density map to get the high quality structure. Okay, so the next one. Okay. So the aim of this, when we want to get the crystal, okay, the crystal. So the aim is to get the big single crystal diffracted at a really high resolution. So in the hope that we get the high resolution, okay, high resolution of protein or resolution of the protein, and then in this case we need to get a very pure proteins, okay, because a very low impurity or very highly pure protein will give a low mosaicity of the data. Okay, so the next one. Okay, so we started when we get the highly purified protein, we straight to the protein crystallization screening. Okay, so firstly we screen on the temperature. Okay, the temperature at 4 degrees Celsius and also the temperature at 10 degrees Celsius. And we also screen for the protein concentrations. Okay, we started with the temperature and protein concentration, and we get these two hits at these two formulations. Okay, at these two formulations. So you can see the crystal is not really good in the shape and quite small. So the next one. And then once we get the hit, we try to optimize the protein crystals. So in this case, the protein crystallization condition is these formulations that we successfully get from the screening. So what we do is that we try to increase the protein concentrations. So the next one. So what we do is we try to increase the protein concentrations and see the results. What happened, what is the effect of protein concentration to the formation of the crystal. So we start with 4 milligrams, 6, 8, 10, and 11 milligram per mil. And as the concentrations get higher, so we get a much more big crystal or larger crystal. And the shape is better, much better compared to the smaller concentrations of the protein. So the next one. All right. Okay, and then we try to optimize further. Okay, optimize further the condition of the protein. So where we dialyze, change the buffer of the samples into this buffer, which is 10 millimolar MOX pH 7 in the presence of 200 millimolar sodium chloride, and then concentrate further. Okay, and we get the 11 milligram per mil. And when we crystallize this time, we get much better appearance of the crystal. With the 3D shape. All right. So the next one. Okay, so then we subjected the protein crystal for X-ray diffraction, and then we analyze the data, and then we get the structure. Okay, so this is the structure that we get. This is the first self structure for nickel CS77 nickel ion hydrogenase. So we get the distal, medial, proximal, and active site determined in this structure as well. So the next one. All right. So when we solve the structure for the CS77, something that we observe that in the proximal cluster, okay, in the proximal cluster, which is near the active sites, so there is the presence of positive peak. Okay, there is a presence of 2 positive peak over here. So this makes us wonder what they are. Okay, so in this case, we try, since we have 2.3 Armstrong, the resolutions that we get for the first structure. So what we do next, because from the 2 peaks, it's really interesting to solve what they are. So what we did is that we try to improve, okay, more the resolution of the proteins, and we're thinking how to do it. And from our observation, from the first crystal structure that we get, so there is the, if I'm not mistaken, this is C-terminal loop. the C-terminal loop that presents in the structure. So what we do is that we try to cut this loop. Okay, so how we do that? So from the Dr. Hafiza's presentation, so Dr. Hafiza expressed the protein without the C-terminal residue. But what I did is that I cut this sequence by introducing trypsin. Okay, introducing the trypsins in the protein. So they cut the this part. All right. So the next one. So next. All right. So this is the purifications. Okay, the after purifications that we did. So this one is without trypsin treatment, and this is with trypsin treatment. So so you can see the purifications after we introduce a trypsin before the purifications. Okay, so resulted in a much pure of the protein compared to the ones that without trypsins. So so this is a very good indicator for us to go further for protein crystallizations. Okay, so the next one. All right. So it's resulted in a very good results where we actually can improve more on the size of the quality of the crystals. And this time we get a larger crystal compared to the previous one. And also the thickness of the crystal is also improved and diffracted at higher resolution. Okay, so the next one. So this is the crystal that we diffracted for the crystals that we finally crystallized from the samples with trypsin treatment. Okay, so this is a measurement for air oxidized state, the next one. And this is for the reduced state. So all the crystals that we use for the diffraction is from the samples that treated with the trypsins. Okay, so yeah, so you can see the patterns of the diffraction spots is very clear, very intense, showing that the crystal lattice in the crystal is well ordered. It has low mosaicity. So the next one. All right, so this is the comparison of the data collections, statistic and refinements that we did, that we get comparing the crystal that we get without trypsin and with trypsins. So it shows that the one that with trypsins, they resulted in high homogeneity of protein samples for crystallization and should improve in the crystal packing. Okay, so the next one. All right, so so we move on with the data analysis that we get from the crystals with trypsin treatment to solve the structure. Okay, so we get the structure in active in the oxidized and reduced form. So this is the difference of the active sites in oxidized and reduced form for hydrogenase. So in oxidized form, there is the presence of oxygen, okay, in the middle of the nickel and ion. And in this case, for active site, we didn't observe any of the ligand. Okay, so the next one. All right, and in terms of the proximal cluster, so there are different observations, different state of the proximal cluster that we observed for the proximal cluster. So this is the in hydrogen reduced form and air oxidized form. So it's confirmed that in air oxidized forms, there are two conformations exist. Okay, in the air oxidized form for proximal cluster iron sulfur. And in the superoxidized forms, they are totally deformed. And okay, so the next one. Okay, so we compare the proximal cluster of the CS77s in the oxidized form with the other oxygen tolerant and standard nickel ion hydrogenase, and we find something interesting. We find out something that much more interesting, because when we compare the proximal cluster, so there is a different. It's quite similar, but there is something in the proximal cluster where we find out this species, this new atoms or new molecule that's attached to this ion in the proximal cluster. And there is no such thing in the oxygen tolerant and standard nickel ion hydrogenase. So this makes us to find out more. So what is the species? Okay, so the next one. So in this case, we also find out the changes, okay, the changes of the structure of the aspartate acids here between the reduced and superoxidized form. So there is a movement of the aspartate 81 and also proximal cluster. Okay. All right. So in this case, we want to determine what is the oxygen species and this W2 species that presents in the superoxidized form. So in this case, we try to identify the stable spin states for each configuration of the oxygen species that might be assigned to this proximal cluster. Okay, so for the O1, we try to we assign these two species, okay, of this molecule, and then so this is the molecule or H2O that assigned to the W2. So we do the calculation by using initial approaches, and also so the next one. Okay, so we come up with the 12th model, okay, the 12th model that from the calculation, and we find out that the model 2 and model 7, it has the most smaller value, okay, of the standard deviations, okay, so it's compared to other models. So this means that this model is much more accurate compared to the other ones. So we conclude that the oxygen one is the hydroxide or OH minus, and the W2 is the H2O. Okay, so this is the results from the initial calculations that we get. Okay, so the next one, and then we do the MD calculations to find out the distributions of the water molecules in the CS77 compared to the standard one. So in this case, you can see that the S77s, they have the glycines 18, and this one they have the histidine 13. So from the MD calculations, we conclude that the formations, okay, the presence of glycines in this position compared to the standard one, which is histidine. So these glycines, so the presence of the glycines helps the water distributions, okay, from the active sites to the outsides, or from the outsides to inside of the molecule of the S77s. So, but in this case, for the standard one, which is highly sensitive hydrogenase. So the water distributions or the pathway of the hydrogen is disrupted by this histidine 13. So there is I can say it's a very special water loop that presents in the S77s. Okay, so this makes the CS77 is very efficient to bring in the water in and out from the enzymes. Okay, so the next slide. So in this case, after we get all the results, okay, from the data analysis, ab initio calculations, and also molecular dynamics simulations, we propose that these S77s, they have a novel molecular mechanisms, okay, for protecting the active site from the oxygen damage. So this is based on the presence of the proximal cluster that has the ability to protect themselves, okay, from the oxygen damage in the presence of the aspartate 81 and also the hydroxides in the proximal cluster. All right, so the next one. Okay, so unlike reported oxygen tolerant hydrogenase, so S77 had a similar mechanism but not identical fashions to deal with oxygen attack compared to other oxygen tolerant hydrogenase. So so the electron supply systems and also the water relocation systems that they have help the S77 to relocate help the water to escape from or restore the deformed cluster. So this is one of the strengths of the S77 to deal with the oxygen attack and becomes not really oxygen sensitive. Okay, so the next, okay, so then, okay, so that is the first project. So the next one is the current work that related to the serine proteases. So this is mostly dealing with the computational approach. Okay, so the next one is the related to the proteases that isolated from Bacillus subtilis strain runt. So runt protease is identified as serine protease that fall under this S8 subtilis family. So it was characterized as thermostable because it able to retain its activity after 30 minutes of incubation in the temperature between 37 to 55 degrees Celsius. And it is also highly stable in organic solvent. And this makes this protease as the one of the candidate that can be applied in various industries. Okay, so the next one. So based from the our observations, okay, from the structure studies of other proteases, it shows that the other proteases, like they, if that thermostable, they have the presence of the cysteine amino acids, cysteine at the calcium loop. Okay, so that is one of the features that differentiates protease, thermostable protease compared to not thermostable ones. Okay, so the next one. So so this so there is the role of the calciums affect the properties of serine protease. So in terms of thermostability, activity, also folding rates of these enzymes. So this cysteine actually displays a strong hydrophobic nature and is also known to stabilize the 3D structure of the proteases. So this is the function of the cysteine, all right, in the calcium coordinating residue in mostly thermostable protease. Okay, so the next one, okay, so compared to the run protease, when we look at these two coordination sites of the calciums, they don't have any cysteine. Okay, they don't have any cysteine in the calcium loop that forming this coordinations, okay, for the calcium 1 and calcium 2 for the RAN proteases. So this is quite different from the other thermostable proteases. Okay, so the next one. So the objective of this study is we try to introduce, okay, what is the effect of the cysteine if we introduce to the RAN protease at the location of the calcium loop. Okay, so we think that by introducing this cysteine at the calcium loop, it will make this RAN protease as a model to see if by introducing the cysteine into the calcium loop, it can actually increase the stability of RAN protease, and we can design the enzymes for other industry. Okay, so the next one. Okay, so this is the structural conformation of the calcium binding loop when we do the simulations. Okay, and then so these amino acids really play a role for the calcium formations, okay, in the RAN proteases. So the next one. So the next slide. And also for the calcium too, also there is amino acids formations that form the calcium loop. The next one. Okay, so the formation of the active sites is also moved with the movement of the calcium amino acids with the amino acids at the binding calcium loop. So we conclude that the binding of the calcium ion has suggested to trigger the sequential concerted movement that affect the geometry of the catalytic site for RAN proteases. Okay, so the next one, we also run so this is just the interactions that we find out between the active site and also the RAN proteases with the substrate. And then we run the molecular dynamic simulation to see the effects of if we substitute the cysteine to each of the amino acids separately. And we found out that it gave a different results where the mutant of the asparagine 183-C give the most stable compared to the original one. So this proved the importance of the calcium coordinating residue to provide the stability of the protein. So there is improvement there when we introduce the calciums. So the next one. And also in terms of the flexibility, so there is a decrease of the flexibility and improve in the packing of the enzyme when we introduce the calcium as well. So the next one. Okay, I think maybe in the 4 slides. Okay, so the next one. And this is also the results from the so this is the last slide where we can see that but when changing the cysteines, so one of the mutants, which is the still N8C, it gives a very good result. Okay, very good result where we can see that they stabilized the protein as well by looking at the active site becomes more buried in the enzymes, and it's maintain the structure and hold the folding of the enzymes. Okay, so the next one. All right, so this is just 2 papers that we published from the studies. Okay, and then the next one. Okay, so this is also related to RAN proteases where we check on the effectiveness of the RAN protease on the leather deherring, and it's seems to show that the run protease is a very good candidate that can be applied in the leather deherring industries. Okay, so the next one. And also other works or potential structural studies that I'm working on now is about the proteolytic activity, the keratinase, okay, the keratinase, and also the Zappa protein, which is we isolate from the Proteus mirabilis. So this is inshallah, two projects that I'm working on now towards structural studies. Okay, so the next slides. Okay, so I want to acknowledge in this opportunity, I want to take this opportunity to acknowledge Prof. Baka and also Prof. Raja and also Prof. Shukuri and also other EMTech members, Dr. Hafiza and also my students. Thank you for the endless supports from them. Okay, so thank you very much.
Okay, thank you, Dr. Dina, for your nice presentation and deep explanation. So, because we have a limited time, I will welcome for the first session to question and answer session. Okay, first I will give a opportunity to Bu Is, please. Please mention your name again and your institution.
Thank you very much, Mbak Linda. Welcome to Indonesia, Dr. Dina and Dr. Hafiza. Yes, I'm Is Helianti. I have several questions. First, it's related to the presentation of Dr. Nur Hafiza. First, in your work, you omitted the tail of the protein, right? Maybe I missed your presentation. What is the reason? Is it because the sequence of detail is not it is not have homologous sequence in database or something else, the reasons? And the second questions maybe I address to Dr. Dina. Thank you very much for the presentation about structural biology related to crystallization. Yeah, I just want to know what is, because further on my knowledge, not all protein can be crystallized, right? So based on your experience, what kind of trait or characters of protein that easily to crystallize and difficult to crystallize? That's my two questions. Thank you.
Okay, thank you very much. I will answer the first question about the C-terminal deletion, because our aim was to crystallize the protein and we've found it difficult to crystallize the enzyme. So we look at the structure of the lipase and we compared with the one available in PDB. So when we superimpose the structure, the one in PDB has no tail at the C-terminal end. So we thought perhaps this tail region is not needed, or it may, if it is present, it may interfere with the crystallization packing and the formation of the crystal growth, the formation of crystal of this 82 lipase. So that is one of the reasons why we removed or we deleted the C-terminal end, and then we managed to improve the stability of the enzyme and managed to crystallize the protein. Yes. Yes, correct.
For example, in our protein also there is long hair, and I needed, but I don't have some reason.
Oh, I see.
In your opinion, by comparing to
Yes, that is one of the ways to delete the flexible region, because in protein usually the flexible regions, especially at the terminal ends, are not function. I mean, it is not functional in terms of the activity, because the activity is in the active site. But it may function. It may responsible for other function of the enzyme. The presentation just now, I mentioned structure guided. So if you have available structure, maybe you can also compare the enzyme structure with other available structures, and you can do mutation from the available information.
Okay, so Dr. Islanti, thank you for questions. So related to the what we call maybe like Dr. Hafiza says, the protein that difficult to be crystallized, right? So your question is related to that one. So from my experience, okay, so from my experience, I think from my reading as well, the protein that difficult to crystallize may be the protein that's difficult to, or they are present in the not really soluble. Okay, they are not soluble in the solution. So if they are not really soluble in solution, so they are really difficult to get crystallized. Yeah, so hydrophobicity. So it has also some of the protein, if they have a very high number of cysteine, okay, cysteine in the amino acids in the sequence. So most probably you have to check it, because so they might be not that stable for protein crystallizations. Yeah. And also I think there is one website that we can actually check the solubility of the protein, whether they are actually can be crystallized or not. So we can check on that. So yeah. All right.
Okay, because maybe one more question. I saw no more question. Okay, sorry. Okay, from chat box we have three. I think this is the final one, yeah, for this question. So for the first speaker, what more important for protein stability enhancement with N-terminal truncated or C-terminal truncated? And what the result of RMSF of C-terminal truncated mutant? What the purpose of mutation of lectin protein sulfose? I think maybe some of parts you have already explained, yeah, but maybe you can more.
Oh, okay. Okay, for the first question, which one is more important for protein stability? I think it is a case by case. It is a case by case condition where you have to look at the structure itself. But I think it is how do I say? If we delete the C, or if we truncate the C terminal end, it will not affect the overall function of the enzyme, because it is at the end, not at the beginning. So if we truncate at the end terminal, maybe there are cases where it can increase the stability, but there is a possibility that we may interrupt the overall structure of the enzyme. For the second question is the result of RMSF. So RMSF in MD simulation is to measure the local flexibility of each residue in the protein sequence. So the higher the RMSF value means the more flexible the residue is, the more movement the more movement of the residue. And for question number 3, what is the purpose of mutating lysine protein surface. You can, okay, what about other amino acid? Yes, for protein structure, usually the hydrophilic residues are located on the protein surface. So you can mutate either lysine or arginine. So it's up to your aim of the work. But in my case, I mutate lysine because lysine more abundant on the protein surface compared to arginine. So we want to see the functional role of this lysine in the stability, as well as hoping that if we mutate some of the lysine surface patches, it will also help to increase the chance to crystallize the protein.
Okay, thank you for your nice answer from Dr. Fiza and Dr. Dina. Thank you. I think I will end this first session seminar. Maybe we, okay. Yeah. I will give this to MC again.
Yeah, thank you so much. Please give applause. Okay, for the first and the second speakers. Before we continue to the third and the fourth speaker, we will give a token of appreciation to the first and the second speakers. And for that, I would like to invite Dr. Ahmad Fatani to give the token of appreciation. Yeah, the token of appreciation will be present to Dr. Yeah, Dr. Nur Hafizah Ahmad Kamarudin. Please give a warm round of applause for our first speaker. Yes. And the next token of appreciation will be present to wait. Yes. Continue. Waiting the Zoom. Yeah, sorry, I'm waiting here. No. I mean like the certificate. Oh yeah, okay. Yes, thank you so much for the first and second speaker, Dr. Nur Hafizah Ahmad Kamarudin and Mrs. Noradina Muhnur PhD. Yeah, everyone, ladies and gentlemen, our moderator is ready for continue our seminar. Yes, without further ado, let's continue our seminar with the third and fourth speakers.
Okay. Thank you, Miss Dea. Okay, I hope you still awake this afternoon. Okay, give applause again. Okay, we still have two more speakers from BRIN. First from Research Center for Applied Microbiology, Dr. Dede Heri Yulianto, and the second speaker, Dr. Joko from Research Center for Genetic Engineering. So for the first presenter is Dr. Dede Heri Yulianto. I will give a brief CV first. Okay, Dr. Dede Heri Yulianto is a senior researcher at Research Center for Applied Microbiology BRIN. He also manager for cryo-EM laboratory BRIN, and his expertise in bioprocess, bioremediation, enzyme and microbial technology, and also now he deep into structural biology. Okay, without any further ado, I will welcome to Dr. Dede to give a presentation. Okay, it's like some technical error. Please wait. Okay. Okay, Dr. Dede will give a lecture about cryogenic electron microscope, accelerates protein structure and engineering research. I think he will also introduce cryo-EM laboratory, BRIN. Okay. Now it's ready, Dr. Dede. Yeah, floor is yours. Yeah.
Good afternoon, ladies and gentlemen. I would like to present about the cryo-EM, how cryo-EM can accelerate the research on protein structure and protein engineering. So I'm from Research Center for Microbiology, Applied Microbiology, and also as a manager of Krios, Tundra, and protein purification. So yeah, this is the discussion about the. This is not for the compare about the X-ray crystallography or cryo-EM, but all the methods is good, but we can each method can complement each other. Yeah. So X-ray crystallography has long been the dominant methods for the using of high resolution protein structure, but recently cryogenic electron microscopy is now catching up, and yeah, this is the a basic method. The differences between the X-ray crystallography and cryo-electron microscopy. So in X-ray, the X-ray beam, we have first to have a crystal sample. So this beam will scatter, and then the diffraction pattern will be detected to the detector, and the detector will make a diffraction pattern and then can be used for the data processing. In case of a cryo-electron microscopy, so it uses an electron beam, and the sample is a protein sample. It doesn't need for crystallization, and then the electron will detect to the detector, yeah, and directly can be used for the data processing imaging. Next, so many researchers say that although X-ray crystallography has been structural biologist-based tools until now, but it also has a major limitation. One of limitation also has been discussed this first speaker. Also, we sometimes difficult to make a protein easily to crystallize. So it takes years to find a way of forming some recalcitrant protein into larger crystal, because X-ray crystallography needs a larger crystal, and then that are suitable for analysis. And also, many fundamentally important molecules is sometimes difficult to be crystal, yeah, or define crystallography. So I would like to introduce you about the story of cryo-EM technique, which is all the researcher awarded as a Nobel laureate in 2017. So Richard Anderson, in 1970, he tried to see the bacteriorhodopsins, but it's difficult and unsuitable for crystallography. There, he tried to use electron microscopy. Therefore, in 1975, he produced the first 3D model of the protein using electron microscopy. And also during the same decade, Joachim Frank, he is a biophysics, and he developed image processing software. And then in 1980, a team by Dubochet, he is also a honorary professor in the University of Lausanne, Swiss. He tried to find a way how the biomolecule can flash freezing in the solution using the liquid ethane. So this kind of technique is basically very important in cryo-electron microscopy. So next, I try to make a very simple technique. So cryo-EM means we talk about the three parts. First, how we prepare the sample using the vitrification under minus 180 degrees Celsius in liquid ethane. So this is a very important step. The other is we try to optimize the electron microscope condition. So this is also a very important step. And the other is the data processing, almost same with the X-ray crystallography. So, but in this step, cryo-EM takes only like 1 hour for 1 or 2 sample preparation. You don't need to make a crystallization, just your pure solution, protein solution, put into Petrobot in 1 hour, and then we run for the electron microscopy for 4 hours. But, and this is the also important step. Maybe more than 4 weeks, you have to develop and refine how you make your crystal fit with your model. Okay, next. So cryo-EM complement traditional structural biology technique. As we can see here, here is the area of X-ray crystallography. So, and this is the another methods of NMR. Usually this is the resolution, 1 Armstrong, 2 Armstrong, and this is the molecular size, molecular weight. So cryo-EM divided into one is a cryotomography, which is the big molecular size, like from 500 kilodalton until 1 megadalton. And this is the method for the single particle analysis. Usually effective for above 100 kilodalton. And for the small molecular protein, we also can still use cryo-EM by using the microelectron diffraction here. But of course, in this part, we need to make our protein in under crystal condition. But we don't need to make a larger or bigger crystal protein, just a very small, like a micron or a nano size of the crystal still can be measured using the micro-ED. Okay, okay, continue. So this is the limitation for each cryo-EM, X-ray crystallography, and also NMR. Of course, cryo-EM also has a disadvantage, like highly specialized equipment. You have to buy the X-ray, cryo-EM equipment, and also expert in the sample preparation. This is very important step in the sample preparation. Sometimes the buffer or your buffer is making a crystal ice in the grid. So it is a very important step to passing the disadvantage of the cryo-EM. Okay, continue. So recently the data entries in PDB is like this. This is the total, the cumulatively data entries in PDB. You can see the blue one is X-ray crystallography. It is still higher because of the total. And you can see that since 2020, the data entries for the protein crystal structure in PDB is also in the log phase. Yeah. And if we see per year annually, the cryo-EM is drastically increased. And it is X-ray crystallography in 2024 is almost twice, yeah, with the cryo-EM. But I think in the future, cryo-EM will be increasingly drastically increased for the data entry in the PDB. Okay, continue. And this is the EMDB electron microscope data bank. So the cumulative number like this, and also this is the per year annually for the structure in EMDB. And the data size, I mean the TD volume size, the data is a variable from the minimum size around 1 megabyte until around 16 gigabyte from the volume for the volume of the we can say apa ya cloud awan molecule ya. Okay, continue. So this is the distribution of the data entry into EMDB. So mostly from 100 megabyte until 1 gigabyte, and also from the 10 mega until 100 megabytes. Okay, continue. And this is also important for all of us. Current status of molecular size entries in EMDB in 2024. So mostly for the single particle analysis, all the technique or the cryo technique entries in EMDB, the molecular size is range from 100 until 250 kilodalton. It is common in the EMDB, but below 100 kilodalton is still rare. So maybe the technique should be also important. Next, and this is the resolution distributed in EMDB, as we can see from the 2005, mostly higher than 15 Armstrong, and very small in the under 3 Armstrong, right? But at the time, the composition is different. Now, the resolution of the below 3 Armstrong is higher composition, and this is a 3 until 4 Armstrong. Okay, continue. So this is the technique in the EMDB. So if we see here that mostly the technique in EMDB is a single particle analysis and tomography data, and also tomograph averaging, and this is a micro-ED. So the technique in cryo-EM mostly enters in the single particle analysis. Okay, continue. So this is one example how cryo-EM could reconstruct the fatty acid synthase structure and given 1.9 Armstrong resolution. So yeah, it is published in 2023. They try to use the SPA and also how they measure the this aryl carrier protein, the important in the fatty acid synthesis in the E. coli. Yeah. Okay, continue. Oh, sorry, in yeast. Yeah. Okay. So I would like to explain to all of you about the cryogenic electron microscope at BRIN. So we have a Krios G4 300 kV. This is the latest generation of Krios, and also Aquilos, Tundra, Talos, and Petrobot. This is for the sample preparation. So the step is we from the sample we can directly, if you want only to see the TEM, you can use the Talos, and or you can use the Pitobot for the sample preparation, and you can analyze this directly to Krios, Tundra, or Talos. Or if you want to do like a cryotomography, then after sample preparation, you go to Aquilos 2 for making the lamella and analysis using the Krios. Okay.
Sorry, Dr. Dede, 5 minutes more.
Yeah, okay. Thank you. We can skip this 5 minutes, yeah? Yeah, okay, okay. Skip aja kan? Maybe everyone already know the cryo facility. This is our cryos. So if we reach 1 Armstrong, then it means that we can see the atomic resolution individual atom. Yeah. Okay, continue. So actually, this cryos can be used for 3 general functions: single particle analysis, cryo-electron tomography, and micro ED. Okay, continue. So this is the workflow for the cryo for the single particle analysis. So your purified sample, then put into a grid sample, and we freezing using Pitobot, and this is the preparation step. And we analyze this using cryo EM and data acquisition. This is for the optimization of the TM, and we obtain the 2D projection, and from here we use the data processing. After getting the 2D projection, we then running with the software, almost same with the X-ray crystallography, maybe like a Cryospark, Reliant, or Phoenix and Code. Yeah. Okay, continue. So how sample preparation will be? Yeah, we can use. Please start. Yeah, this is we present how the sample be prepared in the cryo-EM. Oh, nggak ada soalnya ya? Ya, ini adalah penyiapan untuk, nah ini glow discharge. Jadi grid, so grid is in the glow discharge using the, to make the grid surface is a hydrophilic. And then this is the important step, how we put the sample into grid. And the grid directly enter to liquid ethane. This is the freezing, and but the sample will not form the crystallization, just amorph condition. In this case, our protein sample still in the native state, yeah. And we could do clipping. And after this, we bring to the kiosk. This is very simple. It takes only 1 hour. Yeah. Okay, continue. So, yeah, this is the step by step. Before we prepare, we try to measure the protein content and preparation for liquid ethane, glow discharge auto grid, put the sample, and clipping. Yeah. Okay, continue. And then we run in the cryo EM. First, we try to open the atlas to screening the good grid. This is one grid. You can see in one grid it contain. This is we call this is a grid square. One grid square, we select the grid square, continue. This one grid square, one grid square, there's so many hole. Yeah, and then we select the hole. Yeah, this one hole. And our protein end up in this hole. Yeah. Okay, and then we also can data acquisition from the hole. Yeah. So let's continue. And this one. This is one of the sample, lacase, around 55 until 70 kilodalton. As we can see, still the protein here. Like the protein, then analysis. We got the 2D projection, and then we run for the 3D reconstruction. Okay, continue. And this is the running data acquisition. Okay. So, for example, this is from the apoperitine. Then we reconstruct using the software. We obtain the 3D reconstruction. Okay. Okay, continue. And the other technique is cryo-electron tomography. This technique is using the two equipment like FIB and also Curios. FIB is for the lamella preparation. From the sample, we put into the grid, and the cryo FIB will making the lamella very thin lamella, and then Curios will data collection. Okay, continue. So yeah, I will skip this one. Okay, yeah, this is the FIB containing the NCM and also the FIB ion. Okay. Yeah, this is the workflow for the cryo-electron tomography. After dropping the cell, we freezing using the board, and we mapping using the FIB and milling, and we obtain a very thin lamella in cell, and also we observing using the Curios. So this is like if you have the protein virus, you have the cell and the other tissue engineering, we can use the high pressure freezing using the cryo lift out. Yeah. Okay. And this is the result for the some technique. This is minus 600 nanometer lamella. We try to obtain 100 until 200 nanometer lamella, because the TEM is need a very thin sample. Okay, continue. And this is one of the example. After we obtain this is the output for the Curios, and this is the data processing using the Inspect 3D or AMIRA. Okay, continue. And the third method is microelectron diffraction. I think this is also good for like crystallography, because it is almost same, but we just need a very small protein, and we directly put into the Curios, and we can see the diffraction of the sample. Yeah, okay, continue. This is continue again. This is the last one. How guideline for us, if we have a big sample like a tissue or cells, you can use the cryo-electron tomography. And this is the expected result for your sample. And for you have a protein above 100 kilodalton, then you can directly use single particle analysis, like using Curios or Tundra. And this is the expected result. You can get the image, and you obtain the 3D structure. If your protein is below 100 kilodalton, for example, 17 kilodalton, you can use crystallization. Then we have to learn how crystallization is by maybe Prof. Shukuri later. And the crystallization then continue to Curios to make the micro-ED. And then this is the expected result. I think, oh yeah, this is like one example of subtomogram averaging. You can see using this Curios, and then you can make a bigger or detailed image using the subtomogram averaging. Like this structure is from this membrane. Also, you can see more detail of your protein or a bacterial membrane. I think That's all. Yeah, thank you so much for your kind.
Thank you very much, Dr. Dede, for your nice presentation. Okay, we will go to the last presenter, Dr. Joko from the Center for Genetic Engineering. First, I will read his CV first. Okay. Yeah. Dr. Joko Febrianto Trinugroho is a junior researcher at Research Center for Genetic Engineering, and she did his PhD in life science research in Imperial College London. And his research interest is protein engineering for cell process, and also genome bioprospecting and comparative genomic of microbe, as well as synthetic biology. In today's seminar, he will talk about the breaking limits, enhancing and modifying vector protein function through protein engineering. I will give your time, Dr. Joko.
Thank you very much for the warm introduction, Belinda. The Honorable Head of EMTech and all the advisors and lecturers, and also the Honorable Head of the Applied Microbiology Center, and all the ladies and gentlemen, thank you very much for this opportunity today. So, yeah, my name is Joko. So, yeah, apologies for the hyperbolic title, but yeah, I'll share some perspectives about protein engineering, also the one that I have done before. Okay, I'll talk about two topics. The first one is engineering cyanobacterial D1 protein of photosystem 2. First of all, I'll like to refresh our memory. This is one of the most notable physiological processes that we already know, which is the photosynthesis, which use light and capture carbon dioxide and water, and then to produce sugar. And in the process, it produces oxygen. And this process consists of two main reactions, the light reactions and the Calvin cycle. If we take out the Calvin cycle, we end up having just the light reactions. Next slide, please. Okay, and the light reaction of the oxygenic photosynthesis consists of four major protein membrane complexes, which are the, okay, I've got the ruler. The photosystem 2, cytochrome B6F, the photosystem 1, and ATP synthase. And if we remove all the three proteins, we got the photosystem 2 complex, which has the critical role in the light reaction because it produces or evolves oxygen. Next slide, please. Okay, so these are the cartoon about the photosystem 2 complex. This is the cyanobacteria and the chloroplast. So there are many subunits that forms the photosystem 2. So I just want to point it out that the D1 and D2 protein is from the reaction center of photosystem 2, while I'll focusing on the D1 protein. So this here that forms the reaction center. Okay, next slide, please. And so why it's D1? Because this is the structure of the photosystem 2, which is have been studied over the decades. Over decades. And why is D1? Because in photosystem 2, there are many subunits, chlorophylls, and so on, and it has the manganese cluster, MnA4CaO5, which is the sites which evolve oxygen, which is called the oxygen evolving complex, and it is critical that it's produce oxygen from water for the splitting. So, and D1 itself, so this is the manganese cluster and the residues that coordinated it. And the next slide, please. So why it's D1? Because the D1 protein coordinates most of the ligands that surrounding the manganese cluster. So which this structure was solved in 2011 by Umina. So as we can see here, so this is the manganese cluster. So the D1 protein coordinates around 7 ligands, while the other 2 ligands come from the other protein, which is called the CP43. Okay, so D1 is considered conserved in cyanobacteria, plants. However, evidence suggests that the D1 has a variations. Next slide, please. So, based on the evolutionary perspective, so D1 can be separated into 2 groups, the typical and atypical D1. So it also forms different groups. So the typical, it's the standard D1 protein that works, that evolves oxygen, that works in photosynthesis. And the atypical D1 lacks the ligands that bind manganese cluster, and presumably it doesn't evolve oxygen. And interestingly, one of the groups, which is the group 1, it's called the chlorophyll F synthase. So it's from variation of D1 protein that is important of producing the chlorophyll F, which is occurred in cyanobacteria only. And also the other type of D1 actually presumably have other function, just like the other, the group 2 expressed during dark. So it might related to the nitrogen fixating bacteria, cyanobacteria. Okay, next slide, please. Okay, so this is the phylogenetic trees of the D1 evolution. Just want to a bit focusing on this structure. So this one group GK, group 1 and group 2, which are the typical D1 that lacks the manganese cluster residues, as you can see here. So it's unable to bind strongly with the manganese cluster, while this is the standard D1, which is the normal D1 protein. And next slide, please. Okay, I'll skip this one because the first speaker already kindly explained about the protein engineering. So the aims of the project is to investigate whether photosystem 2 can be evolved to oxidize water using a different configuration of the manganese cluster. So we want to try to do metagenesis in the residue surrounding the manganese cluster of the D1 site to investigate whether it could still evolve oxygen or not. So the strategies are using the site saturation mutagenesis of the ligands. We target single residue or multiple residues using the degenerate and NK codons, and then creating mutants library and then screening. Next slide, please. Yeah, this is the workflow. So we did with inverse PCR to generate the saturation mutants, and then performing cyanobacterial transformation, and then proceed to analysis. And then next slide, please. So yeah, so we use the Synecosis 6803 because it's a cyanobacteria model organisms. Why we choose this organism? Because first, it's been sequenced, it's easy to transform, it's transformable, and it's naturally competent. So we don't need to make a component cell like E. coli, so it's already able to accept any DNA, fragment DNA. So, and then I use this plasmid, which called PRD1031, which integrates to the chromosome. So it's not a self-replicating, but this integration factor goes into the genome of the Synecosis, and this is will integrate to the VSBA2, which is the D1 gene. And then, so the background strain that I use is the D1 knockout strain, which lost the D1 protein. So the idea is if we put the D1 into the knockout D1, so it restores the ability to do photosynthesis, so it could grow phototrophic. Next slide, please. And this is the mutagenesis. So we use the inverse PCR using the NN codon, and so it's amplifying all the plasmid, and afterwards doing a treatment, and then equivalent transformation, do plasmid isolation and sequencing, and lastly, do cyanobacterial transformation. Okay, next slide. Okay, after transforming to cyanobacteria, the next step is to test whether the cyanobacteria has taken all the plasmids, which is by segregating check test or it's called genotyping. So the idea is if the cyanobacteria has the normal D1, so it will have a larger fragment because it's inserted with the kanamycin resistance and the protein itself. So the size of the amplified PCR will be higher, as we can see here. So all of the mutants have fully segregated, meaning that the genome has already taken all the transformed fragment, comparing to the background strain, which is the knockout. Okay, then the next one is the libraries that have been created. What the single residue is E189. We got 3 libraries, and 2 out of 3 that grow phototrophic. So which is good indication. And then this is the 2 residues, and 4 of the 3 residues mutation. Okay, this is for the phenotype analysis of the E189 P, because previously we didn't select for phototrophic growth. So to select for phototrophic growth, we just grow in the media without glucose. So if it's grow without glucose, it means that it could photosynthesize. So this is slightly smear, so it's not clear whether it could grow phototrophic or not, because we don't grow it long enough. But yeah, for the next one. But however, all the other residues, the 2 ligands, cannot grow phototrophic. So it means that the photosynthetic activity is perturbed. And then the 3 ligands also has the same result. Next slide, please. So the same result goes to the 3 ligands. So where they cannot grow phototrophic, meaning that they cannot do photosynthesis. Next slide. And then we also test for the photosynthetic activity by this measurement. So it's called a flash-induced fluorescence. Basically, it's to measure the kinetics of the photosynthetic activity. In short, so if we compare to the wild type, it should have the same profile. So if the photosynthesis is active, so it should have the relatively same profile of the wild type. So just short, so all of the 2 residues mutation and 3 residues mutation, they don't have active PS2. But interestingly, the E189P, it shows some activity, but we didn't have time to repeat and do another experiment. But it would be nice to do probably like oxygen evolution measurement or other stuff as well. And then next slide. So to conclude, synocystis mutants expressing the E189L and K grow phototrophically, while the E189P may be able to evolve oxygen and grow photoautotrophically. But however, it's still unclear how more or residues mutation affect the photosystem assembly and activity. So we need to have more screening, more experiments, more analysis. And third one, PCR using the NNK codon is a fast and simple method for the site-specific random mutagenesis. Okay, this all the colleagues and my supervisors. And okay, next slide, please. Okay, the second one will be just like a perspective. So this are engineering enzymes for polyethylene triacylate degradation. Okay, so this is the team. So I now joined the research group, protein and microbial engineering for good industrial processes. So look at all the happy, look at the smile, all happy family. Okay, thank you. Next slide. So, okay, so now we already know that we are facing the environmental issues, which comes from the plastic waste. whilst the contribution of the PET is the most because it's in 2019 it is the most produced plastics in the world. So it will pose a hazard because as we know, every day we use PET, plastic bottle, food packaging, any household appliances now also almost all have PET on it. So there's the chemical structure of the PET, which is the terephthalic acid and ethylene glycol, so it's polyethylene terephthalate. So we need to, sorry, next slide. Okay, so the current methods for treating the PET waste mainly by incineration and chemical degradation. However, these two methods are not environmentally friendly and even pose hazard because it produces hazard waste and products. So we need to find a way, alternative way to have a more safe and environmentally friendly approach. So there's an alternative called bioremediation by using the enzyme that is produced by microbes, such as fungi, bacteria that could degrade this plastic, the PET, into its monomer. Next slide, please. So, previous studies have reported many sources of PET degrading enzyme from bacteria, such as the cutinase from the metagenomic leaf branch compost, or it's called LC cutinase, the PTase from the bacterium HR29, or it's called BHR PTase, and also PTase from Ideonella sequensis, IS PTase, and cutinase from Thermobifida fusca. So it's many. There will be many more that we have not been explored, possibly. But this currently they are more studied, and then many attempts of engineering has been done by using this protein for the templates. And next slide, please. And to consider treating the enormous waste, we need to have a large scale application. So we need to have an enzyme, PET degrading enzyme that is workable for the industrial application. So there are some consideration for implementing it. The first one is temperature, because we know that the glass transition temperature of PET is around 60 to 70 degrees, meaning that we need to have an enzyme with a high thermostability that works around this temperature. And the second one is the fact that PET could recrystallize. So we need to have an enzyme which is high efficient enough. And the third one is the yield of depolymerization, because over time the enzyme activity could deteriorate due to instability or product inhibition. Because of many degradation, it will inhibit the enzyme. So we need to enhance the stability in highly concentrated PET. And for the last one is enzyme availability, because the instability, low expression of the host could limit the upscaling process. So we need to find an industrial expression host to obtain the large production. Next slide. Okay, so how to get that done? Well, it will be tricky. So we'll struggle, but there are two main routes that might possibly could go to that site. The first one is using the available PTAs that is already in the public. So we know that there are many PTAs or cutinase that is been researched. So we could improve it by engineering, and then do testing and validation. And if it works, we do implementation or upscaling. But if it's still not promising enough, we repeat the engineering process until we get what we desired. And the second one is exploring the novel enzyme by genomics or metagenomics, maybe, because we still have many bacteria. That majority of bacteria are unculturable, so we still have many places that we could find, probably like thermophilics, thermophilic environment, something like that, and do testing, or we could do engineering first, and then the same goes the same cycle until we get the best possible enzyme for upscaling. Can yeah, this is last slide. So yeah, this is our work, the previous and our current works. We are doing engineering PTS from the ideal sciences by computational bioinformatics approach. So we try to improve the thermostability because this enzyme is good, but it has a low thermostability. So we want to improve its thermostability. The second one is we are also doing the novel mining that PTS from the metagenic database, because we have a lot of database now, and like in the CBI, there are many sequence archive, like from shotgun metagenomic data. We could explore it to find a suitable candidate for the PTS. And third one, exploring and screening novel PTS enzyme from Indonesian bacteria. Where yeah, we do have a good biodiversity. We have some bacterial repository that we haven't been explored enough, so it'll be interesting to find out. And the last one, yeah, it's kind of like we got a grant on it. This type of structural studies of engineered PT using the cryo-EM. I think that's all from me. Thank you very much.
Okay, thank you very much, Dr. Joko, for your nice presentation. Okay, now we will go to Q&A session for this last session. So maybe one question, because of time restriction, I will invite for maybe only one question. Okay, there's any from the chat box? There's any question from? Okay. Any from the floor? Okay. Dr. Is, please, one question for Dr. Is. Yeah, so how is the progress of our cryo-EM?
Okay, it is a good question. So actually, cryo-EM technique is easy to be operated, but not easy to be prepared your sample from the customer. It is important because many of the proposal ajuan in the ELSA is sometimes not suitable for the methods. First, sometimes the protein is not enough for the concentration, and it is very important to prepare the pure protein first. Then it easily can be run in the cryo-EM. So until now, last year we obtained 3 structure of the protein enzyme. This year, until this June, we have so many images, but it is still run for the data processing. Yeah, and the data processing already finished is around 5. But should be refinement and validation. It is an important step for the submitted into EMDB or PDB. Yeah, it is very important step. No, if you provide sample 10, we can run in 2 days or 3 days for the data collection. Then we can run directly because we have 4 GPU, 4 we call for GPU, and then we run into different state, we can directly use it. Our limitation is sample. Please provide sample. Yeah, please provide sample. Yeah. If we have a more sample, we can run, because as I present in this slide, only 1 hour for the preparation, only 4 hours for data collection, and only 5 hours. Then more than maybe 1 month, I don't know, have the experience about the data processing to refinement and validation. This is important. I think the most important is to provide your sample. Yeah, provide your sample. Yeah, because the running in chaos is no much time. Yeah, no more time. Only 5 hours. Yeah, a day or overnight. Yeah. I thank you. The preferred concentration is from 1 until 5 or 7 milligram per ml. Milligram per ml. 1 to 7 or 1 to 5. Yeah. Depend on the size of the protein. Yeah. Usually, sometimes in the only 1 milligram per ml, it is enough to see the distribution. Yeah. And the good one is, this is the technical in the lab. We have to keep the protein stable condition, because sometimes they making the aggregation. When it aggregated, it is difficult to be imaged in the KUM. Yeah.
Okay, thank you very much for the question. Actually, probably probably could answer, help me answer, because it is we got a grant but in this structural studies by BRIN and RIM and LPDP. So basically, we want to first, because we do have a KUM, we want to have a look at the structure of the engineered PTs, but probably initially we want to resolve the structure, but afterwards probably we want to do some more mutation like engineering to improve the activity, especially and the thermal stability. Yes, yeah, because I mean, because the structure is already available, so first one, probably what will the effect of the mutation will affect to the structure, and then if we could study that, probably we could help us to guide what kind of mutation that will destabilize, and so we could prevent the destabilizing mutation and prefer the stabilizing mutation, probably like that, something like that.
Okay, I think it's okay. You one question or okay? Two. Oh, Dr. Dini.
Okay, it's about the EDS, the microcrystals. So, what is the mechanism to shoot the crystal, the microcrystal? Because from my understanding, the microcrystal will be flow through a streamline, right? And I want to know how much of the amount of microcrystals that needed for diffractions using EDS in your facilities. Yeah.
Okay. I forgot to put in the slide, but it is a very good question. So in the technically, you can put your crystal very thin, not big. It is different with the crystallography, right? You have to bigger crystal, but in this micro-LED, you only like a nano crystal. So therefore, we have to like digerus ya. And then we have the very small crystal. Then we just put into the grid directly, and we freezing using the liquid ethane, and then we put in the cryos, and then we use electron diffraction. Then only less than 1 hour, maybe less than 1 hour for the optimization. Like SPA, we need 5 hours, but microED less than 1 hour. We directly obtain the electron diffraction. Then I think the next process is almost same with the crystallography. We need a CCP4 for the data processing, and Phoenix and code, and yeah, that's all. I think we can more collaborate for this. Yeah, this more easy. But if you have a bigger protein, I think it's better to use a solution. Yeah, using the SPA. Yeah. Thank you so much.
Okay, I think it's already the end for our seminar today. But before, please allow me to highlight some points from today's seminar. And though we can do protein engineering with like traditional one, directed evolution or random mutagenesis, but I think the knowledge of the structure itself will accelerate to do this to engineer the protein, and then we can do more fasten the process to make them to give them added value for the industrial application. And for this structure biology advancements now coming up with the cryo-EM is already, I think it's accelerate also this research in protein engineering. And but actually this cryo-EM crystallization and then NMR, of course, all actually have a pros and cons, and then we need to know the purpose of our it depends on the molecule that we want to elucidate the structure. And then actually the key of all this method is actually the sample preparation. Okay, I think it's we need to provide the good sample and then let the process run, and then and I think analysis also the harder part, because it's like take months, of course. Okay, I would like again thank you for all the presenters today, Dr. Fiza, Dr. Dina, Dr. Dede, and Dr. Joko. Thank you for all nice presentations. Okay, please give applause. Okay, thank you. I will give part for MC.
Yeah, thank you very much, Dr. Linda, as our moderator today. Yeah, as an appreciation for the insightful information, knowledge, and also experience, we will present award of certificate to the third and fourth speakers, Dr. Dede Heri Yulianto and Dr. Joko Febrianto Trinugroho, and will be given by Dr. Ahmad Fatoni. Okay. First, award certificate for Dr. Dede Heri Yulianto. Please give applause. Yeah, the second certificate is for Dr. Joko Febrianto Trinugroho. Yeah, please give applause. Yeah, thank you so much for all speakers. Dr. Nur Hafizah Ahmad Kamarudin, Miss Nordina Nur PhD, also Dr. Dede Heri Yulianto, and then Dr. Joko Febrianto Trinugroho. Ladies and gentlemen, I know you want to still get more knowledge and information for other speakers, but sorry, we only have 4 speakers for today. So if you want to get another knowledge or information, see you on the next meeting, yeah? Yeah. Excellencies, ladies and gentlemen, we have come to the end of our agenda. Thank you very much to all participants for your active participation. To close today's meeting, please allow me to read a pantun. Buah nanas, buah ceri. Kurang kompak. Sorry. Buah nanas, buah ceri. Dicampur gula, enak sekali. Cukup sekian pertemuan kali ini. Semoga bisa bertemu lagi kemudian hari. I am Dea Mala. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
We have
wabarakatuh.
group photo session here or outside? Here, inside the room. All of participants? Yes, for all of participants, we have group photo session. So please come forward.
Ya, terima kasih Bapak Ibu semuanya. Jadi sebenarnya kita sedang menginisiasi juga untuk skema DPR ya. Jadi tadi pagi sudah kita discuss juga dengan UPM bahwa UPM itu bisa 100% stay in Indonesia ya. Tapi juga bisa certain time juga di UPM. Jadi nanti kalau terutama adik-adik atau nanti Bapak Ibu PI yang punya keadaan di Bogor nggak di Indonesia, kita bisa semoga tahun depan lah kita bisa register dengan daftar ke UPM ya. Karena di sini banyak sekali apprentice yang di struktur dan tadi di sini juga Dr. Hafizah juga still available actually. Yeah, so we can invite her to be a visiting sister also, yeah, together with us. But because Dr. Dina already hired by Dr. Nick, so we cannot approach her anymore. But for Dr. Faiza and other colleagues from UPM still available, so we can actually invite him for especially visiting sister. Yeah. And we may also, if not this year, next year, select some PI from cell biology, 6 maximum, a group of 6. We can send to UPM to train for protein crystallization, especially. Yeah, but we will see later on. Yeah, so we will very selective because maximum 6. Okay, thank you everyone. Yeah. Assalamualaikum warahmatullahi wabarakatuh.
Yeah, for all participants, please come forward because we have group photo session.
Ayo mari Bapak Ibu kita berfoto dulu, terus pulang sore ya.